23 pairs of unreplicated DNA. I'd have 46 of these chromosomes. Okay, so this whole thing here is a chromosome. And that's what I have in like G1, right? I have these. I have unreplicated DNA. Well, now I go into the S phase, right? So in the S phase, I'm replicating my DNA. So now I copy this DNA. Well, hey, guess what? This whole thing here, also a chromosome. And I think that's where a lot of confusion comes in. Okay, so replicated or not, it is still a chromosome. Okay, now once it's replicated though, now I might get one of these, I'm going to get these chromatids. And the chromatids are the duplicated, the copies, right? These here are sister chromatids. Right? So that makes these guys down on the bottom sister chromatids. They're the same copies of the chromosome. And when these split apart in anaphase, during anaphase of mitosis, okay, now it goes back, it's still a chromosome. Okay, so you get these sister chromatids when the DNA has been replicated. So on replicated chromosomes, you're going to have sister chromatids. And those sister chromatids are going to be held together by this part right here. That thing right there, holding those sister chromatids together. Well, that is the centromere. It's in the center. Okay, and that holds all of those, it holds those replicated chromosomes together, it holds those sister chromatids together. You may also have fibers, you may also have proteins, okay, that will run the length of the chromatids and hold them together. And those are called cohesins. And these are the proteins that are going to hold the length of the chromosome together. Okay, so let's kind of pause for a minute and take a look at some of these. So what I have here is replicated chromosomes. So the whole thing is a chromosome. The whole thing is a chromosome. Okay, when they split apart in mitosis during anaphase, chromosome, chromosome. Okay, so the whole thing is a chromosome, whether it's replicated or not. Okay, the sister chromatids, these are down here at the bottom. Okay, or up here at the top, those are basically the duplicates. Those are the sister chromatids. Remember, the centromere is what's going to hold them together right there in the middle. The centromere also has what's called a kinetochore associated with it. And the kinetochore is what's actually going to attach to the spindle fibers. It's made of proteins. And like I said, it's going to actually attach to the spindle fibers so that when separation happens, the chromosomes will pull apart. And then remember, it may also have these cohesions, those proteins that run the length of the chromatid. Okay, so you can see here, it's not really held together by cohesions. There's a pretty good separation there between the two sister chromatids. But on, if I can pull it up real quick, maybe. But on this picture here, okay, you can see how these chromatids here are touching, because those are held together by those cohesions. Okay, so those are, I said those are held together by those cohesions, and so those chromatids are nice and tight with one another. So again, this would be a chromosome. This is a chromosome. Still a chromosome, still a chromosome. Okay. So part of what we call the chromosome dance is that I think that this looks like a person that's standing there waving their arms around, which when you do our dance is going to be you. So if I had this, right, here's my little hand. You guys know that I'm an awesome artist, right? Okay, there's my little foot. Okay. So one side here would be a chromosome, right? One arm, one leg, that's my chromosome. Okay. Well, I'm going to replicate that now. So now I get still a chromosome, right? Yay, we're happy. Doing the chromosome dance. Okay. My arms are sister chromatids, right? My legs are sister chromosomes. Okay, chromatids, sorry. My arms are sister chromatids, my legs are sister chromatids. Okay? This whole thing is a chromosome. Okay? The whole body is a chromosome. Then I get to wear my waist here then. So my trunk here is a centromere, right? And on the centromere, I get to wear this really cool belt. And this really cool belt's like a utility belt, right? I get to wear this really cool belt. Well, that really cool belt is going to be my kinetochore because it's got places where I can actually attach. So it's right there next to my centromere. And it's that protein structure right there next to the centromere, but it's where the spindle fibers will actually be attached. So I get to wear this really cool superhero utility belt around those. Okay? Only thing I'm missing here are my cohesins, which I could have, right? Okay? If I brought my arms real tight up against my ears, if I stood with my legs real close together, okay, I could be held together with those cohesins. So those are all, those are the bulk of your C words, right? Your chromosomes, chromatids, cohesions, centromeres. Like I said, this is the hardest part of the unit is sorting all that stuff out. Okay, so let's look at our actual phases of mitosis now. Okay, so I like this picture here because I think it's a pretty good overview of what happens. But also I just want to kind of remind you of the different phases of mitosis. So we've got prophase, what's called prometaphase, which when you guys were freshmen we kind of just lumped in together with prophase. Metaphase, anaphase, telophase, and then we have to have cytokinesis happen. So let's take each one of those phase by phase. Okay, so during prophase, we're going to have a number of things happen during prophase. So during prophase is when these chromosomes are going to really start to form. Remember, the chromosomes are just the condensing of the chromatin. So we see that loose form of DNA, that chromatin form of DNA, start to wind up nice and tight into the chromosomes. The nucleoli will disappear. Okay, remember that has a lot to do with, there's a lot of high RNA concentration there in the nucleoli. So the nucleoli will disappear. Okay, mitotic spindles are going to start to form. So the spindles will start to lay down asters. I'm skipping ahead of myself here with what I'm going to write. Asters are these kind of like star-shaped structures here. Okay, and sometimes the aster, depending upon the kind of cell it is, the aster will have they will have centrioles associated with them. So the mitotic spindles form these asters, and where they're forming them from is what's called the centrosome. So, yay, we've added another C word to our list. Okay, so the centrosome is where these centrosomes are going to be these areas that are going to migrate to the opposite ends of the cell, and they're going to be extending these microtubules or these spindle fibers. Remember, the spindle fibers are like the roads that the chromosomes are going to follow as they're moving from end to end. So at the end of prophase, then, we start to get into what's called prometaphase. And so during prometaphase, the nuclear envelope is going to start to break apart, okay, or fragment. It's not going to necessarily break down and dissolve because you're actually going to put it back together again at the end. but it's going to break into little pieces and it's going to fragment apart. Have to be able to get that DNA out of the nucleus. So the nuclear envelope will start to fragment, okay, and then those centrosomes, where those asters were, okay, so those centrosomes where you're laying down those spindle tubules, they're going to start to move to the opposite ends of the cell. Those microtubules, those spindle fibers, are going to be extending down into the broken nucleus, into the fragmented nucleus, and they're going to be attaching to those kinetochores. So they're going to be hooking onto that utility belt. Okay, so it's going to hook onto that utility belt that's around that centromere so that movement can occur. So the next step we're going to have here then is metaphase. Okay, so remember it's PMAT, prophase, metaphase, anaphase, telophase. So now it can be a PPMAT, prophase, prometaphase, metaphase, anaphase, telophase. So metaphase is the longest phase. And what's happening here is that those chromosomes are going to line up along the plate, along the equator of the cell. So they're going to line up on the metaphase plate. This will be one of your favorites to be able to recognize under the microscope, because they just line up right there in the middle. And this is done because of those microtubules attaching to the kinetochores. And basically they'll kind of tug and pull and move them around until they get lined up on this metaphase plate. Okay, so on after metaphase, the cell is going to head into anaphase. So during anaphase, those if there are any of those cohesin proteins, then they need to be broken down. It's not a difficult word to spell. So any of those cohesion proteins, they need to be broken down so that the sister chromatids can separate from one another. Okay, and the chromosomes then will start to be pulled towards the opposite cells, the opposite poles. They're going to be pulled towards those centrosomes. So this is where we're actually splitting up the DNA. That's why on that graph we saw before, at the very beginning, where we looked at the different amounts of DNA during the different parts of the cell cycle, it would finally, near the end of anaphase, it would drop back down to each having one copy. So these the microtubules, remember they're attached here at these kinetochores, and these cohesion proteins have been broken, and so they're able to pull the chromosomes apart back towards the opposite ends, so that when a new nucleus forms around them, it will have the right number of chromosomes on each side, and it won't kind of catch the ones that maybe it shouldn't catch, that should belong in the other cell. So the very last phase is going to be telophase. Okay, and so telophase is going to kind of be the opposite of prophase. So we're going to reform the nuclear envelope, and you're going to reform two of them, okay, one around each set of DNA. The chromosomes will actually start to uncoil and unwind back into chromatin so that transcription and stuff can take place. Those spindle fibers will get broken down. They will disappear because they are no longer needed anymore. And we're going to start the process of cytokinesis. And cytokinesis is that division of the actual cytoplasm. So cytokinesis is going to be the division, like I said, the division of the cytoplasm, splitting up those organelles amongst the two new daughter cells. And it's going to be a little bit different depending upon the kind of cell that it is. So what we have over here on the left is an animal cell. And animal cells form what's called the cleavage furrow, because animal cells do not have a cell wall. Okay, so the protein, there's essentially a belt almost of actin protein, which is one of those motor proteins. And so the animal cell, that actin protein, will essentially pull tight. Okay, and as it pulls tight, it forms that cleavage furrow there. And eventually it will pinch it so much into the middle that it pinch into two different cells. But again,